Do you know embryonic cells, they interact with each other to generate the embryo. During the process of differentiation, a cell is committed to develop into a particular cell type. Today's video we are going to discuss about what is commitment, what are types and mode of commitment. If you want to know more about it, then please do watch my video till the end. Hey everyone, welcome back to BIOSKUL. I am Gayatri and today's topic is cell specification and commitment with solved CSIR UGC NET questions. What is cell specification? During the process of differentiation, cells are committed to develop into a particular cell type. So, that commitments are divided into 2 steps and 3 modes. So, what are the steps of commitment? There are the 2 steps of commitment. The first one is the specification and the second one is determination. So, now, what is the specification? The fate of a cell or tissue is said to be specified when it is capable of differentiating autonomously when placed in a neutral environment such as petri dish or test tube. And the specification is the type of commitment which is capable of being reversed. That is, specification is the reversible type of commitment. If a cell is committed to develop into a particular cell fate that is not fixed, it will be reversed based on the environmental condition. That type of commitment is known as specification. Okay. So now what is determination? Determination is a cell or tissue is said to be determined when it is capable of differentiating autonomously when placed into another region of embryo. Here the commitment is fixed. If a cell is determined to become a particular cell type, then it will definitely form that cell type whatever the condition it may be. Okay. So this type of commitment is irreversible. Okay. Now what are modes of commitment? There are 3 modes of commitment. What are those? The first one is autonomous, second one is conditional and third one is syncytial mode of commitment. Now we will discuss one by one. Now the autonomous specification. Auto means the self. So here the cell develops into a particular fate without interacting with its neighbor. The cell fate is determined by its own cytoplasmic determinant. Okay. And this type of specification is seen in invertebrates. And autonomous specification leads to mosaic embryo formation. In the mosaic embryo development, the cell fate is determined by its cytoplasmic determinant and it does not depend on the position of the embryo or its neighboring cells. And the potency of autonomous specification is equal to its original fate. Potency means the ability to form different cell type as it depends on its own cytoplasmic determinants. So the potency is equal to original fate. Okay. So here you have to remember what is autonomous specification, where it is seen, and autonomous specification leads to which type of embryo, and its potency is equal or more than the original fate. Okay. So here in the diagram, as you can see that autonomous specification in early tunicate embryo. Okay. When the 4 blastomere pairs, here the 4 blastomere pairs of 8 cell embryo, they dissociated from each other. Okay. But after dissociation, each forms the structure that would be formed if it would remain in the embryo. Like this one. So A42 and B42, they form the ectoderms, and A41 it forms the endoderm, B41 it forms the mesoderm. And here you can also observe after the dissociation of the blastomere, as its cell fate is the depends on its own. So the A41 it developed the ectoderm, and B42 also the ectoderm. A41 it developed the notochord, and B41 it developed the muscles on the mesenchyma. So here what happened in autonomous specification, the cells or the cell fate is determined by its own. So now comes to the conditional specification. In conditional specification, when a cell fate depends upon paracrine factor or morphogen released from the neighboring cell, not its own. So in the conditional specification, the cell fate is depends on the paracrine factor or morphogen that are released from the neighboring cell, not its own. Okay. And this type of specification is the characteristics of vertebrates and some invertebrates. This type of specification leads to regulative development. So, in the regulative development, the development of embryo, it depends on the surrounding environment. Okay. The surrounding environment, it regulates the development of embryo. So, it's called as the regulative development. And here the potency or the ability to differentiate it into different cells is higher than the original fate. Okay. Now the example. What a cell becomes depend on its position in the embryo. Okay. In the conditional specification, what the cell it would be, it depends on its the position in the embryo. Its fate is determined by interaction with the neighboring cells. Okay. So here it is the normal back cell. Okay. Normal back cell from the blastula. If there is the no transplantation, then it will be developed into the back tissue. But if we transplant the normal back cell into belly region, then cells will form belly tissue. Okay. So what happens here, the back cell develops into the belly tissue because of the interaction with the neighboring cells. If in the second diagram or the V, if the cells are removed from the embryo, the remaining cells can regulate and compensate for the missing part. If we remove a cell from the blastula, okay, then what will happen, the remaining cells, they will regulate and compensate for the missing part, okay, because they are interacting with each other. But in the autonomous specification, if we remove a particular cell fate, then the remaining cells, they cannot compensate for that missing part. Okay. So now the example of gastrula. Okay. So in the early gastrula, the presumptive neural ectoderm was taken and it was placed in presumptive epidermis region. Then what happened? Then it forms the epidermis. Okay, it forms the epidermis. So here the cell fate is determined by its surrounding cell. So which type of specification is it? Is the conditional specification. Okay, and the cell fate here, it depends on the neighboring cells, and the early gastrula are the specified one. So now the late gastrula. In late gastrula, what happens? Similarly, the neural ectoderm, it was taken and placed into presumptive epidermis region. But what it forms? It forms the neural plate instead of the epidermis. So the late gastrula are determined, like here the neural ectoderm, it is determined, like means it will form the neural ectoderm whatever the condition it may be. Okay, and the cell fate depends on its own, and late gastrula, it shows autonomous specification. Okay, Now comes to the syncytial specification. This specification is the combination of both autonomous and conditional specification. So what is syncytium? Syncytium refers to the nuclear division without cell division, resulting in cytoplasm with many nuclei. Okay. So as there is no boundary in the syncytium, the morphogens can affect the nuclei in a concentration dependent fashion. Initially, there is no rigid cell fate for a particular nuclei, but after the cellularization, the conditional specification it comes to play. And this type of specification mostly seen in insects. Okay. So here is the diagram. In the diagram, first what will happen, the egg after the fertilization, the nuclear division will be takes place. As you see in the diagram is the syncytium condition. Syncytium condition means the division of nucleus without cell division. So in the cytoplasm many nuclei are present. Okay. As there is no cell boundary, it depends on the concentration of morphogen gradient. But after the cellularization, what will happen? The conditional specification comes into play. Okay. If the bicoid protein, if the concentration of bicoid protein is higher, then that region it will form the head. In the center region, which is in the light green in color, here the concentration of bicoid protein is less and some nanoprotein are also present. So this one will be from the thorax region, and in the posterior region there is no bicoid protein, only the nanoproteins are present, and that region it will develop the tension region. So now the previous year CSIR UGC NET questions. With respect to development of any organism, autonomous specification would result in which type of development? Autonomous specification result in which type of development? It will develop into mosaic embryo. The second question. Following are certain statements regarding morphogen gradients and cell specification. Okay. So let's see what are the options. A. Morphogens are always transcription factors. Morphogens are transcription factors but not always. So this one is wrong. Option B. Morphogens can be paracrine factors that are produced in one group of cells and travel to another population of cells. Yes, the morphogens are paracrine factors. They are produced from one group of cells and they can travel to another population of cells. C. Option C. When the concentration of morphogen drops below a certain threshold, cells stop differentiating and never get determined to another fact. No, it's wrong. Option D. Morphogen gradients are involved in conditional specification. Yes, it's true. Morphogen gradients are involved in conditional specification. So which combination of the above statement is true? The option B and option D is true. So here the option 2 is the correct answer. Now the question number 3. Which of the inference given below would you draw from the following tissues transplantation experiments performed with the early and late gastrula stage of newt? Prospective neuron of host region, then the prospective epidermis of donor region. Then what will happen? Differentiation of donor tissue. Then the donor tissue it differentiated into epidermis. We have discussed in the early gastrula, if neuron is taken from one tissue and it is placed in the region which become the prospective epidermis, then the neural tissue it becomes the epidermis. Then the second is the prospective epidermis is taken from the host region and is placed in the prospective neurons, then it becomes the neuron. Similarly, in the late gastrula, prospective neuron of host region is placed in the prospective epidermis. Then it becomes the neuron, not the epidermis, because here it is the autonomous specification and the cell fate is determined in the late gastrula. Prospective epidermis, host region, prospective neuron in donor region, then the donor tissue is differentiated into the epidermis. So what is the question? Cells of early nude gastrula exhibit conditional development. Yes, it exhibit the conditional development because the cell fate is determined by the neighboring cells. Second option. Cells of nude gastrula exhibit autonomous development. No. Cells of late nude gastrula exhibit conditional development. No. Cells of late gastrula exhibit autonomous development. Yes. So the early gastrula it exhibit conditional development and late gastrula it exhibit autonomous development. So the correct inferences are P and S. So the option A is the correct answer. Question number 4. If you remove a set of cells from an early embryo, you observe that adult organism lack the structure that would have been produced from those cells. Therefore, the organism seems to have undergone. Okay, if you remove a cell from the early embryo and you observe that the adult organism lack that structure. Okay, so it will be the autonomous specification is the correct answer. If it would be the conditional specification, then what will happen? The neighboring cells they interact with each other and form the lost part. But here the organism lack the lost part. Okay, so it is the autonomous specification. Question number 5. The ability of cells to achieve their respective fates by interacting with other cells is known as. If a cell interact with other cell to achieve their fate, then that one is known as the conditional specification. Option B is the correct answer. Question number 6. When the blastomere pairs of 8 cell stage tunicate embryo are dissociated, each forms of the structure it would have formed had it remained in the embryo. However, the notochord and nervous system get specified only if different blastomeres get the chance to interact. Given below are certain interpretations formulated from the above results. So here there are two options. The first one is that when the blastomere pair of 8 cell stage tunicate embryo are dissociated, each form the structure what it would be formed if remained in the embryo. Okay, is the one. And the second one is that the notochord and nervous system get specified if they get chance to interact with different blastomeres. Okay, there are the two options. So here, so below are certain interpretation formulated from the above two results. Okay, option P. Each pair of blastomeres forming respective structure indicates autonomous specification. Yes, it's autonomous specification because they will form the structure what it would be if it will remain in the embryo. Each pair of blastomere forming respective structure indicate conditional specification. No. The notochord and nervous system development indicate autonomous specification. No. Because here it is written that notochord and nervous system get specified only if different blastomere get the chance to interact. Means if they get interact with the different blastomeres or the neighboring cells. So that is the conditional specification. The notochord and nervous system development indicate conditional specification. Yes, is the conditional specification. Which combination of interpretation is most appropriate? Is the P and S. Where is P and S? Is the option number C. Thank you so much for watching my video till the end, and my upcoming video it will be on induction and competence. It's very important topic in view of CSIR NET, so please stay tuned. Bye bye.